In today's lecture, we'll be covering gene expression, specifically taking nucleic acid language of a specific gene and using that to produce a specific sequence of amino acids, so protein production. The process involves two steps, which are termed transcription and translation. The below diagram shows the large ribosomal subunit of a prokaryotic cell. Our RNA is shown in gray, and proteins associated with this subunit are shown in yellow. The active site is actually revealed by the green inhibitor in the middle. So this is revealing the actual active cleft or active site of this large ribosomal subunit. Now, just to describe the process of protein production, in eukaryotic cells, we first undergo transcription, where a specific gene is used to code for a specific mRNA molecule. So ultimately we take a specific gene, specific sequence of nucleotides, and a copy of that gene is made in the form of mRNA. mRNA is then processed and exported out of the nucleus in eukaryotic cells, and then is picked up by ribosomes, and also with tRNAs and other important factors will lead to translation of the mRNA into proteins. So this is a two-step process. The first step being production of mRNA is called transcription, and the production of a polypeptide chain is the translation portion. Keep in mind, prokaryotes are, this process is highly conserved between prokaryotes and eukaryotes, and we still have transcription and translation in simpler organisms such as prokaryotes. However, there isn't a separation between the two events due to the lack of the nucleus. So in eukaryotes, we do get a separation between transcription and translation due to the fact that eukaryotic cells have a nucleus. Now, there are different types of RNA that are involved in the transcription translation process. mRNA, which stands for messenger RNA, is essentially a copy of a specific gene that is going to be used to serve as instructions for building the protein. So it will code for the specific polypeptide chain of a protein. rRNAs are also an important component of the protein building process. They are, R stands for ribosomal RNAs, and they are components of ribosomes. And in fact, rRNA ultimately performs most of the major functions of the ribosome, such as forming pockets for DNA binding as well as peptide bond formation. Then there are also tRNAs involved in transcription, in translation, and they are called transfer RNAs, and essentially they match up the correct amino acid to the correct anticodon on an mRNA molecule. And then we also have a variety of other types of RNA molecules, such as snRNAs, and these are small nuclear RNAs, and they have different processes such as splicing. The snRNAs, which are small nucleolar RNAs, are used to help process and modify rRNAs. And then we have also other types of what are called non-coding RNAs that perform various functions other than in the protein synthesis process. This is just a bacterial ribosome RNA molecule, and you can see our RNA, although it is a single-stranded nucleic acid, it can fold in different regions to form a more two-dimensional structure, which is actually critical for different RNA's functions, such as forming a two-dimensional structure to form a tRNA versus two-dimensional structures that are needed to form binding pockets for tRNA on ribosomes, etc. And you can see that they form these two-dimensional shapes due to interaction between components of the same single-stranded molecule, so they can actually form base bonding between regions of the strand. And in this particular structure here, this purple region is just highlighting a really important active site of this ribosomal RNA, this specific bacterial ribosomal RNA. Now, the process of transcription involves the use of a very important enzyme called RNA polymerase. And RNA polymerase is ultimately going to first have to bind and recognize the beginning of a gene, and then ultimately help open the gene up so that the two strands base pairs are revealed, and then complementary pair ribonucleic acids with the DNA strand to build a complementary RNA strand. So you can see it has some important features that allow it to bind and then perform RNA synthesis. It has special clamps that help clamp down on the DNA. It also has the ability, as with some accessory proteins, to open up the DNA, melt it to ultimately generate what's called bubble formation. And it will then allow for or then synthesize the mRNA molecule, which is shown here. So it has the ability to complementary pair the ribonucleosides to the DNA strand and then ligate them together, so peptide bond phosphodiester linkage, and ultimately form the mRNA molecule, and it has a special channel in which that will exit. So ribonucleosides will come in to a channel to allow for complementary pair formation, and then this will then be further ligated to form the phosphodiester bond and form that single stranded of mRNA as it comes out. So the bubble that's formed by RNA polymerase is about 35 bases wide, and then you can see here there's about roughly 9 pairs that are forming of the RNA molecule with the DNA strand, and then it will start to fall off on the back end as the RNA polymerase moves forward. So the job of actually making RNA, specifically mRNA for transcription, is the job of RNA polymerase. There's also what are called transcription factors that are critical to also helping stabilize the structure and enhance transcription rates, which we'll talk about a little bit more later. Now, when we think about the whole transcription process, we're taking a sequence of nucleotides and that are written in the DNA molecule within the gene, and essentially RNA polymerase is going to make a complementary copy of one of the strands, which is the 3 to 5 prime strand of the DNA, and build an mRNA strand in the 5 to 3 prime direction. So what you're seeing here is the DNA, and on the other side is the newly made RNA strand that's being synthesized, so the mRNA. And individual bases will be placed that are complementary to the base in the DNA, and the 3' hydroxyl group of the sugar that's already been added at the base above will attack the phosphate group on the new nucleotide, which will help lead to hydrolysis of a pyrophosphate, and that energy will help drive peptide bond formation between those two nucleotides to allow for ligation of the strand. So let's go through the basic steps of transcription. And the easiest way to go through the basic steps of transcription is to go through prokaryotic transcription, because these same basic steps are also seen in eukaryotes, but they have just an additional layer of complexity. So when we think about prokaryotic transcription, there are 3 basic stages or steps. The first being initiation, the second being elongation, and the third being termination. And this particular slide actually covers initiation of prokaryotic transcription. And ultimately, we start with a specific gene, and obviously we need RNA polymerase to start the transcription process. And so what you're seeing here is the core enzyme, which is RNA polymerase in prokaryotes, which has 5 subunits, will interact with the DNA. An interaction with the DNA for RNA polymerase requires, in most cases, what's called a sigma factor in prokaryotes. So the sigma factor will interact with RNA polymerase and help lead to binding of RNA polymerase to the specific promoter region of a gene. And the promoter region of the gene, which you're going to see in another slide in a couple seconds, is the promoter region contains specific conserved sequences that allow for binding of the machinery to the start of a gene. So ultimately it will help recruit the RNA polymerase to that site so that then transcription can occur at that initiation start site from that point forward. And you'll see that the sigma factor allows for or increases binding of RNA polymerase to the promoter, and then ultimately RNA polymerase will then start its copying of that gene sequence in the form of mRNA. Now, RNA polymerase will bind at the promoter with the sigma factor. Then this will lead to opening of the DNA molecule by what's called, they think, melting of the DNA. And then this opening will reveal the two strands of DNA and allow for RNA polymerase to then build a complementary strand of mRNA at that bubble site. Once the mRNA molecule starts to be synthesized, the sigma factor in most cases is lost, and RNA polymerase will continue forward. And this is supposed to mark a lot of times the beginning of what's called elongation. And an elongation is where the mRNA will continue to be synthesized in that 5 to 3' direction until termination. And termination, obviously, is when the process is completed and mRNA is finished, the RNA polymerase has finished completion of the mRNA molecule, and then the DNA will fold back up. And we'll talk about this in just a second. So when we think about something as basic as a promoter, promoters are specific regions usually above the start site in which RNA polymerase will start to make a copy of the DNA molecule that allows for binding of RNA polymerase to that DNA molecule. And the promoter usually contains what are called conserved or consensus sequences that really help allow for binding of the machinery. And this particular promoter here is a bacterial promoter of a specific gene in E. coli. And you can see that there are two important conserved sequences in this particular gene. In prokaryotes, most prokaryotic genes have what's called a -10 sequence gene, which is also called the TATA sequence and also called the PRIB-NOW box. And another important element that's seen in prokaryotic genes is also what's called the -35 element, which you can see here as well. And you can see the -35 is given that name because it's close to -35 bases upstream of the actual start site. And the TATA sequence, which is also called the PRIB-NOW box, is at the close approximately around the -10 sequence. And this is usually where we'll see those particular consensus sequence in prokaryotic genes. So ultimately, these two conserved sites are going to be helpful to recruiting RNA polymerase to this location so that transcription can proceed forward. Now, eukaryotes also have specific sequences. However, these sequences vary from gene to gene, so there's a little bit more variability as far as conservation of sequences, and there's different types of conserved sequences found in different types of genes. So it's not as simple as thinking about there just being a -35 element and a -10, the TATA box or PRIB-NOW box. It's a little bit more complicated, and there's different types of conserved sequences found in different genes in eukaryotes. So obviously this promoter region is going to be very important for the initiation. Now remember, I told you that we have initiation, which is where RNA polymerase is recruited to that site, and ultimately the first few nucleotides will be laid down of the mRNA molecule. And elongation is where the mRNA molecule is continued to be synthesized by RNA polymerase, and then we reach our final step, which is termination. Termination is thought to be initiated in bacteria by either what's called a Rho factor protein, which will help lead to disassembly of this complex, or it also could be due to what's called a terminator sequence. Some prokaryotic genes have a terminator sequence that's a special sequence that actually will form more of a hairpin-like shape that will actually cause RNA polymerase to fall off the gene, which will then allow the DNA to refold and the mRNA molecule to be released. So we reach that termination process of prokaryotic gene transcription. So keep in mind, eukaryotes is very similar to this process with an initiation, elongation, termination, but there are some subtle differences in promoter sequences, different elements that are involved, etc. So now let's look a little bit more in the complexity of eukaryotic gene transcription. And essentially, we have multiple RNA polymerases, unlike prokaryotes. Prokaryotes have one. And we actually have what is listed here as 5 different RNA polymerases. And the first 3 being obviously the most common here. We have what's called RNA polymerase 1, which is thought to synthesize larger RNAs. And we have RNA polymerase 2, which has been one of the most importantly characterized because it actually synthesizes mRNA molecules. So this is going to be responsible for making our mRNA transcripts that are going to be used to make proteins, as well as other types of RNA. And then we have RNA polymerase 3, which transcribes most tRNAs and some of the rRNAs and small RNAs. And then we have RNA polymerase 4 and 5, which we see in plants only, and those make siRNAs. So like I said, eukaryotic transcription is a little bit more complex. We obviously all still have promoters upstream of our gene that are ultimately going to be critical for recruiting RNA polymerase. And this shows you actually 3 different genes, eukaryotic genes, and their specific promoter region, or what's called a core promoter elements. And core promoter elements are, the basic machinery, regulatory domains that are going to be needed to produce transcription. And you can see there's a certain version of a TATA box that's found in each of these 3 genes, which are highlighted in green. So we also have something like a TATA box at a certain position, which you can see is about 24 to 32 bases upstream from the initiation site. And the initiation site is shown here in kind of pinkish colored here, so that's the start site. Now, not all eukaryotic genes have TATA boxes. Others have other types of core promoter elements like the DPE, which is actually downstream of the start site, and others as well, such as the BRE, the MTE, the XCPEI. So we have different conserved consensus sequences that help recruit the machinery. So we have a greater variety of what are called core promoter elements that are needed for transcription of particular genes. So initiation of transcription by RNA polymerase 2 at the promoter site of a gene requires what are called general transcription factors. And these general transcription factors are shown here. TF2D, TF2A, TF2B, etc. And they're thought to bind in a particular order to really help recruit RNA polymerase 2. So the first one to bind is thought to be transcription factor 2D, and it binds at what's called the TATA binding site, TATA box, and you can see it has a special subunit that's called TBP, which is thought to be a TATA binding protein. And then other factors will then interact with TF2D, which is A and B, and ultimately that will then be joined by RNA polymerase 2 and transcription factor F. Now, transcription factor E and H, which are joined after, which you can see here, E is thought to have an important enzymatic property to help melt DNA, and the H has helicase activity, so that's ultimately going to help open up the DNA molecule as well. Now, in addition to general transcription factors being required for initiation of transcription by RNA polymerase 2 for production of mRNA, there are also what are called gene-specific factors. And these gene-specific factors bind to regulatory sequences that are upstream or outside of the promoter region, and they are thought to help enhance the stability of the RNA polymerase and lead to enhanced transcription rates of a particular gene. And so this allows eukaryotic cells to have control over what gene-specific factors are actually produced at a given time, which will then help lead to enhanced or decreased transcription rates of a particular gene at a certain time or point in the cell cycle, etc. And you can see this particular gene transcription factor will interact with this complex, help stabilize it, and then essentially this will then allow transcription to occur. Now, not only do some genes require a single gene regulatory protein, but some actually require multiple regulatory proteins that bind to specific regulatory sequences on the DNA. We call these regulatory sequences a lot of times enhancers if that specific gene regulatory protein binds and enhances transcription. So sometimes specific genes may require multiple regulatory proteins to allow for transcription to occur. And many molecular biologists actually spend quite a bit of time studying what important elements are found in what's called the core promoters, such as something like the Tata box that's required for transcription, and also what other regulatory sequences upstream or downstream are also critical to transcription. And they can identify those by different deletion experiments, etc. And we'll talk a little bit more about this at the end of the lecture. So initiation of transcription, once you have all those general transcription factors and gene-specific transcription factors, and we have RNA polymerase there and eukaryotic genes, there are important features of RNA polymerase that are really going to help allow for elongation to start so that we get continuous production of an mRNA chain, and also, as you're going to see, it will also help coordinate processing of the mRNA molecule as it's being made. So RNA polymerase has a really important portion to its structure, and it has what's called a carboxyl terminal domain, which is CTD, that sticks off of RNA polymerase. And this particular C-terminal domain has heptad repeats, and these heptad repeats actually have serine residues that can be phosphorylated. And these phosphorylation sites actually allow for important functional aspects of RNA polymerase. So phosphorylation is actually catalyzed by not only transcription factor 2H, but other kinases as well. And this helps trigger separation of the transcriptional machinery. And these phosphorylated sites also help allow for collaboration and interaction with different enzymes that are needed for RNA processing. So when we think about mRNA, remember in eukaryotes, mRNA is transcribed in the nucleus, and before it actually is exported out of the nucleus, it needs to be processed or modified. So the three modifications that happen to eukaryotic mRNA is that the 5' end will get a cap, and it will get what's called a methylguanosine cap. The 3' end will end up getting what's called a poly-A tail, which is multiple adenosine residues are added to the end. And then also we get removal of the introns, and exons are ligated back together, and this is accomplished by the spliceosome. Now, to start with just the capping, this particular diagram over here kind of describes the important capping process. And the capping process is accomplished by an enzyme that actually has two different kind of catalytic domains. This enzyme has the ability to, first of all, remove the the phosphate group at the end, and then also add the guanine or guanosine containing residue to the 5' end. So the first active site will actually serve to remove that pyrophosphate, and then the second one will actually add the special guanosine residue to the 5' end, which you can see here. And then a methyltransferase will then add a methyl group to the guanosine residue at that 5' end. So that 5' end at the end of this processing step will have what's called the methylguanosine cap, a guanosine residue added in what's called a 5' to 5' linkage with a special methyl group. So it helps mark it as the beginning of the transcript. There's also another enzyme called the poly-A polymerase, which is going to add the adenosine residues to the very end of the 3' end to produce the poly-A tail. And the poly-A polymerase will add something like 200 to 250 adenosine residues to the very end of the chain, which will form that poly-A tail. So we have a special 5' guanosine cap and then also a 3' poly-A tail at the very end. And then the third processing step for eukaryotic mRNAs is removal of introns and ligation of exons. So this is what's called splicing of the mRNA molecule. Now splicing is done by what's called the spliceosome. And the spliceosome, this complex of many different proteins in actually RNA molecules, will actually recognize specific non-coding regions called introns in the mRNA molecule. And if you remember from some of your previous classes, introns are specific portions of the mRNA molecule that will not be translated into a protein product. These are non-coding regions and they need to be excised, and the actual coding regions, which are the exons, will then be ligated together and then used to produce a polypeptide chain. Now, the spliceosome recognizes where an intron begins and where it ends based on certain sequences, conserved sequences. And the nucleotides that you see here in red are the ones that are highly conserved that will ultimately are used as indication points for where the intron starts and where it ends. The ones in black are not as highly conserved, but are usually what are seen at those positions. So that's going to help indicate the beginning and end of an intron for the spliceosome. And this particular diagram shows the order in which the spliceosome assembles and helps excise that intron and ligate the exons back together. So ultimately, the pre-mRNA is going to require what are called small RNAs, which are also snRNAs, and also what are called SNRPs, which are small nucleoproteins. So ultimately, we have different combinations in proteins and RNAs that are going to form the spliceosome. You can see that there's a particular order. So ultimately, U1, which is shown here, will interact with the start of an intron based on the sequences that I showed you on the other slide. And then you also have joining of to this particular complex, the U2. The U2 actually recognizes an important adenosine residue, and ultimately that is going to then an adenosine, nucleotide will then ultimately recognize and pop one of those adenosine nucleotides out, which will help form the loop formation later. And then you're joined by other subunits, which you can see here, which is ultimately going to allow for interaction with each other and also formation of the loop or lariat formation. Then that will help mediate excision of that intron and ligation of those exons back together again. Now remember how I told you that transcription and processing of the mRNA is somewhat coordinated together, and this is due to that C-terminal domain of RNA polymerase. And this is showing you the C-terminal domain and its key phosphorylation points by the different kinases, as well as some elongation factors, but you'll also notice that the C-terminal domain and its phosphorylated sites also help serve as docking points for the processing machinery such as the spliceosome and capping enzymes. So you can see these capping enzymes are going to automatically bind to certain regions of this tail domain and start the capping process, as well as the spliceosome. So the ultimately processing of the mRNA will start to occur as soon as that mRNA starts to be made by RNA polymerase. And remember, the important kind of features of the C-terminal domain is that there's this heptad repeat, which contains serine residues, which will get phosphorylated and serve as kind of key binding sites for that machinery. Now, once we've completed mRNA and that initiation, elongation, termination event involving RNA polymerase, obviously that sequence of nucleotides will then need to be decoded or interpreted into a chain of a chain of amino acids. And this is known as what's called the genetic code. And the genetic code is almost universal between prokaryotes to eukaryotes. And basically it defines what are called base triplets that serve as codes for amino acids, and some of which you would have already learned in some of your previous undergraduate molecular and cell biology courses. And what you can see is you can determine what particular amino acid a base triplet serves by looking at a certain codon, such as U, and UUC, etc. You can see ultimately what amino acid that base triplet codes for. There are 64 possible codons. If you take all the 4 different nucleotides and you put them in different combinations of 3s, you would get 64 different base triplet codons. And ultimately, we have only 20 amino acids we use to build proteins. So there's more than one codon usually for each specific amino acid, which you can see in this particular chart. The job of matching the correct codon base triplet with the correct amino acid during the translation process is done by tRNAs. So tRNAs have a really important structure, and the fact that they have one end at the 3' end, which a specific amino acid will be attached to, and then they have at the opposite end what's called an anticodon region. And this will base pair with a particular codon in an mRNA molecules to help deliver that amino acid for assembly into a polypeptide chain. So ultimately, a tRNA forms what's called kind of a cloverleaf shape to form these important anticodon and also stem sections or the particular 3' end that's going to allow for binding of an amino acid. Ribosomes are also another important type of machinery that's involved in the translation process. So tRNAs, and now we have ribosomes. Ribosomes have 2 subunits, a small and a large, and both eukaryotes and prokaryotes have ribosomes, and they both also have a small and large subunit. In prokaryotes, the small is called the 30S, and the large is called the 50S, which you can see here. So the structure of the final ribosomal complex is shown here or modeled here. Some important features of ribosomes are the fact that they have 3 binding sites for tRNAs, designated the A, P, and E site. You can see in this particular diagram it's designating where those sites would be. The A stands for aminoacyl site, the P, the peptidyl site, and the E, the exit site. And there's also a kind of channel that ultimately those amino acid residues are going to be linked up in a chain, so the polypeptide chain will kind of exit through that channel. And there's also an important what's called peptidyl transfer center, which is the enzymatic catalytic site for peptide bond formation by the ribosome between 2 amino acids that are carried by adjacent tRNAs. So it's going to help stimulate peptide bond formation between amino acids. tRNAs, remember, their job is ultimately to match up the correct codon with their anticodon region of the tRNA so that it brings in the correct amino acid. Now, there is an idea that's thought that tRNAs do have some flexibility in interaction with specific codons, and this is what's called the Wobble interaction. And how this flexibility works is that the 3rd position of the codon can sometimes be mispaired. And this is called the Wobble interaction. So you can see in this particular scenario it's correct, and then in that 3rd position it actually doesn't have the correct pairing, A with U. However, the other 2 positions are correctly complementary paired. This allows some flexibility such that the same tRNA molecule can then be used to deliver an amino acid for multiple codons, because we know that a lot of times more than one codon specifies for a certain amino acid, and so that same tRNA molecule would be used potentially to deliver an amino acid even though that 3rd position doesn't complementary pair. Now, the job of matching up amino acids with the correct tRNA is the job of aminoacyl-tRNA synthetases, and they are able to match up the correct amino acid with the correct tRNA because they have a special binding site for the tRNA that will recognize the anticodon region, which is shown here in this model of a specific aminoacyl-tRNA and its tRNA. and also a binding site for the acceptor stem so that it can match up the correct amino acid or join it at that particular point. So energy used from the hydrolysis of ATP is utilized to add the amino acid to transfer the amino acid to the tRNA molecule. And when we add an amino acid to tRNA molecules in our cells, this actually leads to what's called charging of the tRNA. So the tRNA is said to be charged. So ultimately our tRNAs are going to be continuously, amino acids continuously added to them by the job of these amino tRNA synthetases. So let's talk about the translation process. So we talked at length about transcription, and then we've also talked about now the machinery needed for translation. So let's talk a little bit about the steps of translation. So translation, initiation, elongation, and termination are still the same terms that we used in transcription that are also utilized to describe the steps of translation. So initiation in bacterial translation is thought to involve the following steps. The first step, binding of the 30S subunit with mRNA at the AUG codon requires what are called initiation factors 1 and 3. So you can see in this diagram the smaller subunit binding to the mRNA and specifically binding at where the AUG start is. And the AUG is the specific codon that helps designate the beginning of translation. And this is thought to be interact with one of the RNA molecules of the small subunit and called the Shine-Delgarno sequence, and that will ultimately interact and help allow for that small subunit to bind to that AUG site. Then you'll have the first initiator tRNA molecule, which is a formal methynyl tRNA. It will associate with the complex, and this involves a factor called IF2 that has bound GTP. So you'll get joining of that tRNA molecule to the small subunit and mRNA. And then finally, the large subunit will join the complex, and this involves hydrolysis of GTP by IF2, and ultimately then that tRNA will enter into that P site of that large subunit. So basically now we have our machinery together. We have our large and small subunit mRNA and our first tRNA and the P site waiting for the second tRNA to come into that next A site. So this is initiation in bacteria or prokaryotes. Now initiation in eukaryotes is a little bit more complex, as you can see. It requires at least 12 initiation factors, and the small subunit is called the 43S instead of the 30S, and it will form as a pre-initiation complex with some other factors such as ELF2 and ELF3, and also the tRNA, the starting tRNA. So we have this small complex with the starting tRNA, and then ultimately that complex will then interact with the 5' end of the mRNA at the cap site, and this is mediated by another protein called ELF3 and another one called ELF4G will help drive the interaction between the cap and this 40S subunit. And once the 43S complex is bound to the cap, it will actually scan forward to reach to the AUG site. And then ELF2 is hydrolyzed, its GTP, etc., and the large subunit joins. So one of the key differences from this complex is the fact that there's more initiation factors involved, and the small subunit with the starter tRNA will actually bind to the cap and then scan forward, which is different than we saw in prokaryotes before that large subunit is joined. This would complete initiation. Now elongation shows the steps of actually making a polypeptide chain of amino acids as the ribosome moves forward and scans the mRNA molecule. And how this happens is due to positioning of the ribosome on specific codons and also the binding of specific tRNAs. So you'll see in this particular diagram here, we have our complex with the large and small subunit and our first tRNA molecule in the P site. The next tRNA will come into what's called the A site, And this will put two tRNAs in close proximity to one another. It will be binding to the next codon of the mRNA strand, and this will allow the ribosome to join these two amino acids that are carried by these two tRNA molecules. So you'll get peptide bond formation between them. Once peptide bond formation occurs, the ribosome will then translocate forward to the next codon. The tRNA, which was the first tRNA, will then move to the E site and it will exit. And the P site tRNA now has two amino acids on it that are linked together by a peptide bond, and it will then be next to a new tRNA that comes into that A site, bringing another amino acid. That amino acid will then be linked together with the other two amino acids carried by the tRNA, and this will continue the chain or production of the polypeptide chain. And then once again, the ribosome will translocate forward 3 nucleotides and ultimately allow for the next tRNA to come in. So ultimately allow for synthesis of that polypeptide chain, one codon at a time. Termination is the final process in the translation step. So we had initiation, elongation, and now termination. And keep in mind, elongation, remember, you're building the polypeptide chain. Well, once that process has been complete, we get what's called termination. And termination is where the ribosome will stop building the polypeptide chain. And this is mediated by what are called stop codons, either UAA, UAG, or UGA. And this requires special release factors. And these release factors in eukaryotes are called ERF1 and ERF3. And they'll work together to recognize the stop codons, and this will actually lead to release of the ribosomal complexes, release of the polypeptide chain, and release of the tRNA, so that the complex all falls apart, and we now have our polypeptide chain and our mRNA molecule, which can actually be used to translate more polypeptide chains. Now, most translation occurs by what are called polyribosomes or polysomes, and this is where multiple ribosomes are actually moving along the mRNA molecule one after another to produce their polypeptide chain. It allows for a much faster process of protein production. So you'll get assembly of one large and small subunit. It will move along as soon as that cap is available again, or the AUG for prokaryotes. Then the assembly of that ribosome will occur again, another one at the beginning of the mRNA, and so you'll have multiple ribosomes at once at different points on the mRNA molecule translating this particular mRNA. And this is something we see in the cytoplasm or also on the rough ER where translation is occurring. Now, just as a summary, we've talked a little bit about a review process of transcription, translation, how we partially regulate this process as far as general transcription factors, etc. Now let's talk about a few techniques that are used to actually study transcription. And this becomes important when you look at other more newer papers in the field, and some of these techniques might be something that you see in your assigned reading. So reporter genes are an extremely useful tool to measure where transcription might be occurring, and this can become really important when just looking at a single cell or actually looking at a developing organism like you see in this particular picture. So this happens to be what's called a transgenic mouse embryo. So it's been a genetically altered embryo where they actually have taken the regulatory region of what's called the myogenin gene, which is a gene that's important in muscle development, and they put it upstream from a bacterial beta-galactosidase gene, which actually serves as a reporter. So all they're taking is actually the promoter and other regulatory sequences and inserting it into this reporter gene plasmid. And where that gene would normally get expressed, you're going to actually express the reporter gene instead. And it works as a great reporter gene because with a different staining step, it will actually turn where that particular gene is being expressed blue. So it serves as a marker as to where transcription is occurring. Now, this is not the only type of reporter gene that's used, but there's other reporter genes as well. There's what's called the luciferase gene, etc., and they all are on specific plasmids that you could actually put the regulatory sequence of a gene in front of and look for expression of that reporter gene in different tissues to really see where that particular gene might be expressed, the timing of that expression, etc., by using that reporter gene as a control. Not only are reporter genes commonly used to look for where a particular gene might be expressed, but scientists also use what are called DNA microarrays to look at levels of transcription of particular genes. And the way microarrays work is you have these plates that these circles on the plates actually have specific DNA for a certain gene in certain circles. And what you do is you'll collect or break open the tissue or the cells, and you collect the mRNA, so what's actually being transcribed in those cells or tissues. You then take that mRNA and you make cDNA out of it, and the cDNA, when it's made to the mRNAs, will actually have a fluorescent tag. And then you can add them to your microarrays. And what this will do is the DNA that's actually being transcribed, which is now cDNA, will bind to the DNA that's on the microarray coordinate, for that particular gene, and it will fluoresce at the genes by binding together. You'll see what genes are actually being expressed by binding of the DNA on the plate to the actual cDNA. And the greater intensity means you have greater expression, or the lack of, if you have no color change, that means that gene is not being expressed in that certain condition. In this particular scenario, you're looking at yeast growing in glucose-rich medium versus growing in ethanol-rich medium. So two different types of medium, and what are the differences as far as expression of certain mRNAs, and what are the same. And the expression is indicated by coloration, the red or the yellow color, which you can see here, fluorescent dye, green or red. And then if both of them are if the same gene's being expressed in both conditions, then you get a combination, which you get yellow. So these DNA microarrays are really great because it actually allows you to test what genes are openly being expressed in certain cell types. So it will give you what's called an expression array of the proteins that are being expressed in that certain condition. Now, these microarrays or DNA microarrays can be utilized even to determine different genes that might be expressed by different types of cancers, which you can see here. So this is showing you human breast cancers that are ER positive and ER negative, and ultimately shows you what genes are being expressed for each and what are being expressed in both gene types. Or both cancer types. Why this is important, because basically a scientist or doctor could identify what genes are being expressed in either cancer or one cancer, etc. And then it will help give that doctor a better understanding as to the genetic expression of that particular cancer, and may even help allow for doctors to specialize what treatment they give to that individual based on what genes are being expressed or not being expressed in that particular tumor. Another important technique for studying gene expression is trying to identify what important elements are in the core promoter, as well as other elements outside of the core promoter that might be critical for transcription of a gene. And so this is showing you here a particular study that looked at how much transcription occurred when certain deletions of those regions were generated. So ultimately you can take a sequence, the promoter region, core region, and other elements, other regulatory regions, you can put it in front of a reporter gene, and you can look expression of your reporter gene. And when you do certain deletions, which are shown here in the black box, here and here, those will either have no effect on transcription rates of that gene, or it may have an effect. So you can see here, when the whole piece of this particular gene is present, you get 100% transcription rate. However, when you remove the Tata box region, you get only 38% transcription of that gene, etc. And you can see here, this last piece, which is in the -30 to -20 region here, it had no impact on transcription rates. So this will help scientists identify important regulatory sequences that might be necessary for transcription of a gene, and ultimately help either review conserved sequences or new regulatory sequences that really give a better understanding as to how that expression of that particular gene is regulated in cells. So some of these you might see as techniques that are utilized by scientists in different supplemental reading or other type of reading you might see in the future.